Hi, my name is Büşra GÜNDOİDU. I'm a master's student at Yıldız Technical University Molecular Biology and Genetic Department. Today I'm going to present our study about anti-diabetic and anti-inflammatory activities of endemic Gypsophila calyx extracts. My presentation consists of introduction, materials and methods, results, discussion and conclusion parts. The Gypsophila is third largest genus of the Caryophyllaceae family. The genus is represented 55 species, of which 33 are endemic in Turkey. This number is equal to almost 50% of the total number of species. Gypsophila species are known as çöven, helva kökü, and sabun otu in different areas of Turkey. Gypsophila species are annual, biennial, or perennial herbaceous plants. Flowers of Gypsophila are small and numerous. Stem and leaves are glabrous or glandular. They have 5 petals whose colors change from white to pink. Gypsophila calyx is a taxon of Gypsophila genus that is a perennial herb. The plant grows on gypsum coasts and steppe of the Iran-Turan region. You can see the Gypsophila calyx plant in picture 1 and the distribution of it in the figure 2. As you can see on the map, it spreads central and eastern Anatolia of Turkey. Gypsophila species are used for food industry. It is used especially in helva making to give the crispy feature, and ice cream, delight, and some local food production. Some of Gypsophila species like Gypsophila calyx are known to have high saponin content. Saponins have foaming properties, so aerocalyx is used to making soap. In addition, they use for manufacture of liqueurs, cough and respiratory system disorders as traditional medicine. Previous studies indicated that Gypsophila species have antiproliferative, antibacterial, antifungal, and antivirus activities. Materials method part. The plant materials. Gypsophila calyx was collected in Iğdır Road, Turkey in June 2017 at 100 meters altitude by Professor Sezgin Çelik and Associate Professor Mustafa Korkmaz. The aerial parts of the collected plant were dried in the shade and stored. The extraction of plant material. The aerial parts of air-dried Gypsophila calyx were ground in the powdered root, a herbal grinding mill, then weighed with a rough balance, and after that, ground and powdered Gypsophila plants macerated in hexane, ethyl acetate, methanol, and methanol water solvents with increasing polarity. Each solvent was macerated 3 times, and the solvents were evaporated after each treatment. The obtained extracts were condensed using a rotary evaporator. The anti-diabetic activity. The anti-diabetic activity was evaluated by the method of Ramakrishna et al. With slight modifications described by Shen et al. 10 microliter of extracts was mixed with 40 microliter of 0.1 molar sodium phosphate buffer and 100 microliter of alpha-glucosidase prepared in buffer. The mixtures were incubated at 25 degrees for 10 minutes. Thereafter, 50 microliter of 5 millimolar PMPG prepared in buffer to the solutions was added. The mixtures were re-incubated and at 25 degrees for 5 minutes, and their absorbance was recorded by reading in the microplate reader before and after incubation at 405 nanometer. Acorbaz was used as standard. The percentage inhibitory activity of the extracts and standard against alpha-glucosidase enzyme were calculated according to the equation. You can see that on the slide. A0 is the absorbance of the control and A1 is the absorbance of the extracts. The anti-inflammatory activity. The anti-inflammatory activity was evaluated by the method of Forestong and Nuktaborn with slight modifications described by Yıldırım et al. 10 microliter at different concentrations of extracts or standard endomedicine were added to 20 microliter ethanol, 20 microliter pure water, 25 microliter of sodium borate buffer solution, followed by addition of 25 microliter of type 5 soybean lipoxygenase solution in buffer. After the mixture, After the mixture was incubated at 25 degrees for 5 minutes, 100 microliter of 0.6 millimolar linoleic acid solution was added, mixed well, and the change in absorbance at 234 nanometer was recorded for 6 minutes. Medicine was used as a reference standard, and the percentage inhibitory activity of the extracts were calculated according to the equation. Statistical analysis. The data were given as means plus or minus standard deviations and analyzed by one-way analysis of variance, followed by the two-case multiple comparison tests using GraphPad Prism. 50 differences between means at p less than 0.05 levels were considered significant. And our results. The extracts displayed moderate and weak antidiabetic activity. I included the inhibition graph of a single dose, 250 microgram per milliliter. The ethyl acetate extract of Gypsophila calyx showed the highest inhibitory activity with values of almost 24%, and it is followed by methanol, hexane, and methanol water extracts. In table 1, we can see the IC50 values of the extracts. The ethyl acetate extract, which has the highest inhibition value, has the smallest IC50 value with 0.539 mg per milliliter. And our anti-inflammatory results. The methanol water extracts showed the highest anti-lipoxygenase activity with almost 87% inhibition value at a concentration of 250 microgram per milliliter. And you can see at table 2, the methanol water extract with an IC50 value of 0.082 mg per milliliter exhibited the highest anti-lipoxygenase activity, and it is followed by ethyl acetate, hexane, and methanol extracts respectively. Discussion part. There is only one report of antidiabetic activity of Gypsophila calyx extracts in the literature. The methanol and water extracts were obtained from aerial parts of Gypsophila calyx. Both extracts display moderate antidiabetic activity. The methanol extracts had an IC50 value of 205.88 microgram per milliliter, and the water extracts had an IC50 value of 235.55 microgram per milliliter. Also, phenolic compounds of the extracts were identified by RP-HPLC. The methanol extracts had 3,4-dihydroxybenzoic acid as main compound, and it is followed by syringic acid, rutin, vanillin, rosmarinic acid, and p-hydroxybenzoic acid. The water extracts had rutin, rosmarinic acid, and syringic acid. In a current study, our study, the antidiabetic activity results of the extracts showed similarity with the previous study, but there are quantitative differences of the IC50 value. The quantitative differences of the IC50 values may be connected to extraction methods and main compounds of the extracts. And conclusion, to the best of our knowledge, this is the first report of the anti-inflammatory activity of Gypsophila calyx extracts. Also, there is no study on the antidiabetic activity of hexane and ethyl acetate extracts of aerial parts of Gypsophila calyx in the literature. The result of the current study showed that Gypsophila calyx has strong anti-inflammatory activity against 5-lipoxygenase enzyme, especially the methanol water extracts exhibited the highest anti-lipoxygenase activity with an IC50 value of 0.082 mg per milliliter, and it is very close to indomedacine used as the control group with an IC50 of 0.22 mg per milliliter. The extracts displayed moderate and weak antidiabetic activity. Further studies are needed to identify and develop active ingredients of the extracts, and actually we are still working on these extracts and develop their active ingredients. It was my presentation. Thank you for listening.